Recording in progress.
Hello, and a warm welcome to everyone. Thank you for joining us. For those of you not familiar with us, Inscopix is a neuroscience tech company in the heart of Silicon Valley, and we provide complete miniature microscope solutions to scientists that want to map brain circuits in freely behaving animals. Before we close out this year, we want to share with the neuroscience community our newest imaging application using Inscopix Miniscope systems. We will be highlighting a new and exciting research application exploring the use of genetically encoded optical sensors to image striatal dopamine dynamics in freely behaving animals. Today's speakers are Dr. Alban de Kerkhoff Daxard and our research scientist, Kevin Zitelli. Dr. de Kerkhoff has an illustrious career trajectory stemming from advanced training in molecular biology, biochemistry, and molecular neurobiology at the University of Leuven in Belgium. After a brief period at the Pasteur Institute in Paris, Dr. de Kerkhoff is now presently the research director for scientific research and the Laboratory of Neurophysiology in the Faculty of Medicine at the Université Libre de Bruxelles in Belgium. His research program focuses on the role of the basal ganglia, their neuronal ensemble activity, and gene expression in neuropsychiatric models and other pathophysiology in the striatum. Dr. de Kerkhoff's group was the first to demonstrate in vivo that medium spiny neurons, or MSNs, in the striatum, the indirect pathway of the basal ganglia, has an inhibitory effect on locomotion and drug preference, and that these are necessary for drug sensitization and the cataplextic effect of antipsychotics with genetic targeting techniques. We are very excited to have him speak more in detail about his latest research efforts today, and we'll hear from him very shortly. Our Inscopix speaker is Kevin Zitelli. Kevin obtained his master's degree at the University of Pittsburgh in 2016, where he studied genetically and functionally distinct circuits of the globus pallidus. Kevin will be talking today about similar research studies using Inscopix miniscopes and optical sensors with the InView system. After joining Inscopix in 2017, he has been a key force for our science team, leading product and application development for the company. We want to thank both Dr. Alban and Kevin for all that they do. Thanks you so much, everyone. So before we get started, I want to make sure everyone attending understands the rules of engagement. We ask that you please provide your questions to us via the Q&A window located on your screen below, and we will do our best to prioritize those questions so that our presenters can ask them at the end of the talks. Please know that we may be unable to address all of the questions depending on the time available. So without further ado, I'm going to turn this over to Dr. de Kerkhoff. Alban, thanks again for joining us today. I'm going to let you navigate the screen share from here.
I hope all of you see my screen.
We do.
Okay, perfect. Thank you very much, Jacqueline, for this nice introduction. And thus today I will present the not yet published result that we obtained in the lab about the fact that striatal encoding of behavior differ between direct and indirect pathway neuron in basal ganglia, in basal property, and dopamine integration. First, I want to start by thanks the people in the lab who are help and working on the subject, Patricia Bonavion, Amandine Cornil, Delphine Ottoman, and Christophe Varin. I will start by a short introduction about basal ganglia, but I imagine that all of you already know what is basal ganglia. Okay. Thus, basal ganglia in mammals are composed by different subcortical nucleus or in nucleus in the midbrain. They are connected in a specific way, and the connection and the organization are the same in human and mouse. And I will use mouse to go a little bit deeper. The fact that the main and entry nucleus from basal ganglia are the striatum, receive mainly glutamatergic afferents from the cortex and from the thalamus, and also midbrain nucleus as substantia nigra pars compacta or VTA. And in the striatum, you have two main striatal projecting neurons. One give rise to what it's called the direct pathway because they project directly to the output nucleus from basal ganglia. And the indirect pathway pass through additional nuclei before to reach the output nucleus. The ventral part of the striatum, it's mainly involved in reward circuit and in disease as drug addiction. And the dorsal part is mainly involved in motor control and learning and involved in Parkinson's disease, as most of you know. Now I will show you a very simplified scheme to illustrate what I want to explain to you, the general connection and the potential role of the two main populations of the striatum. As I previously said, the fact that striatum receive glutamatergic afferents coming from the cortex and the thalamus, activating them. They receive also dopaminergic afferences, working differently on both populations following the dopamine receptor express. And in the striatum, you have mainly these two projecting neuronal populations. The first one, expressing specifically dopamine D1 receptor, is giving rise to the direct pathway, and has a GABAergic project neuron. They inhibit the cells in the outside nucleus, substantia nigra pars reticulata or internal globus pallidus. And the net effect of inhibiting a GABAergic neuron have a net effect on thalamus as activation. Next, thalamus as glutamatergic neuron activate cortex. Now, the other pathway, indirect pathway, composed by also GABAergic projecting neurons, expressing dopamine D2 and adenosine A2A, projecting on external globus pallidus, also composed by GABAergic neuron. Thus, the net effect on the last nucleus was subthalamic nucleus, composed by glutamatergic. This is the only glutamatergic nucleus in the basal ganglia. Thus, activation of neuron in the output nucleus. Thus, if you activate GABAergic neuron, you have inhibition on the thalamus and on the cortex. Thus, this is how the model predict the potential role of these both populations, but this is a model. Now, it is important to demonstrate that in vivo is indeed the case. And the first approach used is what I call causal approach, meaning that you try to disturb one pathway or the other one to see what is the consequence on the behavior and locomotion. Alors, indeed, we was the first to demonstrate, thanks to our new A2A Cray mice, that if you get rid of the indirect pathway neuron, although the technique is easy, we express the diphtheria toxin receptor, you inject diphtheria toxin in the striatum, and as you can see, you have no more indirect pathway neurons. And the consequences, this is the control. In an open field, you clearly see that you have activity of locomotion, but decreasing during the 1 hour of recording. But without indirect pathway neuron, you have a clear hyperlocomotion, demonstrating that indeed these neurons have an inhibitory effect on locomotion. 1 year later, the group of Anatol Kretzer with Alexei Kravitz, using another method, is optogenetic stimulation of the direct or indirect pathway neuron. They showed that by activating direct pathway, you have an increase of locomotion. And in the case of activation of the D2 or indirect pathway, you decrease locomotion. An additional way to demonstrate that is come from the Greengard with Hélène Bhatea by specifically deleted a very important phosphatase called DART32, was mandatory for the signaling in this SPN. And you see that when you kill the DART32 in indirect pathway, we have hyperlocomotion, as we have done here. And when you inactivate specifically in direct pathway, you induce hypolocomotion. And 1 year later, we demonstrate also with ablation of the indirect pathway, we have the same result. Thus, all these results indicate that direct striatal projecting neuron has increased locomotion or have a positive effect on locomotion, and the indirect striatal projecting neuron inhibit or the locomotion. Later, people use another approach. It's correlative approach, meaning that we record, the idea is to record both populations during a behavior to see what is the encoding properties of the both population. And the first result and the first example is the paper from the team from Rui Costa with Guang Kui, by using fiber photometry in the striatum and using calcium binding protein sensor to record the transient of fluorescence. They show that during moving behavior, turning to the right or turning, they show that both populations are activated to the onset of the movement. The same group, 3 years later, using no fiber photometry but miniscopes, recording are able to record specific neuron in the field of interest by using specific analysis with matrix. They show by first compare pair of behavior following the distance of this behavior, and after to see compare the neuronal activation similarity with behavior, and they demonstrate that in fact they have a strong and significant correlation between neuronal similarity and behavioral similarity. That means that in fact same kind of behavior give rise to the same kind of neuronal activation. And you see that they obtain that in both population, direct or indirect. And here the difference is that the moving, specific moving behavior or all behavior that they identified in 14 clusters. If they compare finally the coefficient correlation between these two populations, they find the same kind of coefficient correlation. The last example that I will present to you is the work from Mark Schnitzer and John Parkhurst, using exactly the same miniscope approach, recording both populations, and you can see that direct or indirect pathway neuron activity depending of the state of the mice. And if we compare both populations, for example, in the case of speed or in the case of time from motion onset or offset, you cannot distinguish. Thus, all these results show that D1 and D2 SPN are co-activate and share highly similar dynamic properties. Thus, finally, how to reconcile the fact that causal approach showing clearly antagonist rule, and in correlative, we don't highlight this difference between D1 and D2 SPN. Thus, we have tried to solve this contradiction by also performing high density recording on both populations during spontaneous locomotion. Our protocol is really simple. We have a mice in an open field. Here, we record during 30 minutes and the activity of both populations. But first, we have identified different behavior. Okay, thus on the video tracking image, using deep lab cut approach, we are able to label 8 different parts of the body. And after 5 feature extraction, we are able to identify 6 physical behavioral features as body speed, head speed, movement angle, body length, neck elongation, and head elevation. After, with unsupervised clustering, we identify different clusters, as you can see here. The next step was to label all this cluster with the full repertoire of spontaneous behavior in an open field that is listed here. And you can see that we are able to reconstruct the different, this 12 behavior during a time of an open field. Next step, here you see the mean of all the session from all the mice, and you can see that during the 30 minutes of open field, the active behavior decrease as expected, and the quiet behavior increase. And here you have the mean between the D1 mice or A2, A3 mice, the repartition of this behavior during 30 minutes. Thus, this result, I hope, convince you that we have a reliable identification of 12 behavior. Next step, it's calcium imaging, okay? And we perform the analysis of the calcium imaging frame by using the Kalmhan pipe to identify spatial components. Here you see raw data from all these identified spatial components, but we are working on deconvolute peaks to perform our analysis. Here is to confirm that indeed we're recording a neuron in the dorsal striatum. First, you can see that we are able to record around 200 neurons by session. And whatever the behavior, you can see that in all sessions from all mice, we have the D1 SPN are more active in a significant way than the indirect pathway neurons. Now, if we split between the 12 behavior, you clearly see that we have a higher activation in the direct pathway neuron in comparison to the indirect one, except with the immobility where the reverse result. Thus, this show indicates that we have probably a difference of behavioral tuning properties between both populations. Next, at the cellular level, we want to compare the activity of neurons during the first 15 minutes of the open field or during the last 15 minutes of the open field. For example, here you can see more a neuron is black, more is active. We compare at the beginning the first half of the behavior of the open field or the last part of this open field. And for example, you see in the straight locomotion that the pattern of activation between neurons are really similar. But to the other side of the spectrum, you see that from the D2 neurons in grooming, you have a clear redistribution of the pattern during the 2 time periods. Thus, it gives no neuronal similarity activation in the 8 behavior, you can see that here again, D1 SPN have a higher neuronal activation similarities. Okay, but the fact that we compare the first time period with the second time period, we can imagine that we have some bias or some problem that we can done, that it could be, for example, the time from recording in each population for re-encoding behavior are different. Or the internal state, for example, novelty, stress, tiredness could be different in both populations. That could be to explain this difference. Oh, okay, I forgot to conclude that indeed D1 are more consistently activate. And to avoid this bias, our first analysis is there. We decide to divide the 30 minutes in 5 bins of 5 minutes in a way to make all possible comparison, time comparison, and you see here all the possibility of comparison, okay? And if you are doing that, here you see what I present to you just in the previous slide. In that kind of comparison, you clearly see the difference of similarity with D1 and D2. But in all comparison, all possibility of comparison, we maintain the same result as this comparison. Now, if we go in the same kind of analysis as previously, this is what we obtained, and I previously show you when we analyze in that way. And if we take the other extreme, it means 5 minutes one, 5 minutes the other one, and so on, we maintain exactly the same difference of higher similarity in the D1 than in the D2 SPN. Thus, the difference in similarity is a state properties of behavior encoding. Now, we use exactly the same analysis that Klaus and Costa have done to compare the behavioral similarity and neuronal activation similarities. And you are not surprised that we obtain same result, meaning that we have a high correlation between neuronal activity similarity and behavioral similarity. But if we do the mean between, we compare the correlation coefficient of neuronal activation and behavior, we find a difference, meaning that we have a lower correlation coefficient from D2 SPN. This seems contradictory or different or a problem with the result from Klaus and Costa, but remind that in our case, we record our neuron during 30 minutes, and in their case, between 10 and 15 minutes. And if we reanalyze our data after 10 minutes of recording, we obtain exactly the same result. Thus, we can conclude for this part that correlation between neuronal similarity and behavioral similarity is higher in direct pathway neurons. Thus, the fact that dopamine is really important for modulate, for example, locomotion. And thus the way of the two populations react to the dopamine, are different. Thus, our idea is to see, okay, dopamine could influence the encoding properties of both populations. To do that, same basal condition that I presented you before, and here the fact that either we inject control condition with saline or amphetamine to increase the concentration of dopamine in the striatum. Here, result with saline injection, control injection, and we are not surprised that again we repeat the result that I presented you previously. But if you make amphetamine injection, you see that you clearly decrease the activation similarity in both populations, showing that neuronal activation similarity in both populations is strongly alleviated after amphetamine administration. But we want also to see if the correlation between the neuronal activation similarity and behavioral similarity, if they are different also after amphetamine treatment. This is the saline condition. We find exactly what it was previously shown by Klaus or here in the lab. If, sorry, thus you can see, we can compare both and no difference. Surprisingly, after amphetamine treatment, finally, we don't lost a correlation in basal condition or after amphetamine treatment. We maintain the same correlation coefficient in both conditions, even if in the previous slide, I'm showing you that we have clearly a decrease of similarity, but not on the correlation. But in the case of dopamine D2 SPN, we have a decrease of the coefficient of correlation, showing that amphetamine only uncoupled neuronal activation and behavior in D2 SPN. Okay, thus I show you that you have clearly a difference, but next step is why don't try to see in a normal condition of our normal behavior, what can be happening in the dopamine release and how the changing of this dopamine release could change the encoding of both populations. And we thought, okay, we have a miniscope was able to record fluorescence in the case of calcium imaging. Why don't use it also to make dopamine sensing? And to do that, we decided to try two different dopamine sensors. The D-Light, as you know, it's composed by the dopamine D1 receptor, was fused with a fluorescence protein in a way that when dopamine binds the dopamine D1 receptor, changing of configuration, and you generate fluorescence. The other one that we test is GrabDA. It's composed with the dopamine D2 receptor, and we use the mutated one, it's called DA2M, with some additional point mutation to obtain a better signal noise ratio. We inject both construction in the dorsal striatum, and we choose to work with GrabDA because we have a higher and better expression in the dorsal striatum. Now, the first result that we obtained with a tight collaboration with Jonathan Zapata from the Inscopics. And here you will see, in that case, the raw data, and here it's the image from delta F on F. First result obtained in dorsal striatum. I showed the movies. When we analyze that during the time, you see that we have transient of fluorescence there, following here the time of 1 minute. If we do the same in the nucleus accumbens, you can see that also you have transient, but from lower at the lower level of fluorescence, but also you can see that the frequency is also decreased. That's not we have to demonstrate, okay, this is fluorescence, but this is related to dopamine. It's not an artifact or I don't know. To demonstrate this aspect, we first use a pharmacological approach. Here, recording in saline condition, you see that we have the same profile that I present to you in the dorsal striatum. First, we inject amphetamine to increase dopamine level. And you see that the signal change is higher, but also the frequency decrease. As a control, we use a specific antagonist against dopamine D1 receptor. And you see that you don't change the profile in saline condition or with an antagonist against dopamine D1. I remember you that GrabDA, it's a dopamine D2 derived dopamine sensor. And when we put aldol, we can clearly see that we lost this transient. But to go a little bit deeper to really convince us that it's really related to dopamine, we use another approach. In that Cre mice expressing specifically Cre recombinase in dopaminergic neuron, we inject an AAV expressing in a Cre-dependent manner red-shifted opsin activating opsin. We inject GrabDA in the nucleus accumbens, and with the green lens, we record what's happening at the level of dopamine signaling, and also we are able to make optostimulation, red optostimulation of the dopamine terminals. Here, to show you that we are indeed in the nucleus accumbens. Here is a movies of one optostimulation, and what you can see, each time that we have an optostimulation, we clearly induce a transient of fluorescence. But I hope that I convince you that the observed transient are dependent of dopamine release. Next step, related to the analysis. Because, okay, I show you analysis about the time. But you said, okay, why you have to use micro endoscope? From do that, I can use cheaper fiber photometry. But our idea there was to say, okay, can we also have information related spatial information, what is not possible to obtain in fiber photometry. I take again the movies for the dopamine release in the dorsal striatum. And we do the analysis with tight collaboration with Srinivas Goru-Sandilya, also from Inscopix. And the first analysis was only to take the higher level of fluorescence. And you can see that during the time, this different frame, you see that the pattern of the high fluorescence moving in the field. To go deeper in the analysis, Srinivas decided to have a cutoff to only taking into account the spike upside of the ratio of delta F on F of 0.05. When he's doing that, I show you here an example of 20 frame. What you see here, it's the beginning of fluorescence. And during the frame, you have an evolution of this fluorescence to that finish here in red. And the whole frame, if we analyze the frame all along the time, you can see that we have different kind of pattern. This one, no real difference between starting fluorescence and the end of the fluorescence. But for example, in that case, you have a small surface of fluorescence who finally spread over the region of interest. I show you that. You have the contrary here from that. And so you see all the different kind of spreading of fluorescence along the space. Srinivas decided also to generate a plot of Y position and X position from starting point to end point of each of these transient. And what we have shown and observe, if we are putting amphetamine, you clearly see that you have a reorganization of this pattern, and also you have an increase of the size of the spot, meaning that the kinetics are decreased. Thus, I hope that these preliminary results indicate that miniscope imaging allow dopamine release with spatial and temporal information. And you can imagine that your ongoing work is to combine calcium imaging of both striatal projecting neuron with imaging of dopamine release during spontaneous locomotion to understand their coupling and rule by using dual color microendoscopy. So before to give the floor to Kevin, I hope that in addition to the fact that it's possible to make science in Brussels, we have additional argument to convince you to visit us in Brussels, even if probably Brussels is a hellhole, but don't worry, it's a joke. Thus, I give the floor now to Kevin.
Sorry about that. Hopefully you should be able to hear me now. Yeah, thanks, Albin. It's great talk and great to see the meticulous work your lab is doing, looking at these causal and correlative approaches to dissecting the basal ganglia circuits. Let me get my screen share up and running. There we go. Okay, and thank you all for joining today. I'm really excited to present the work we've been doing imaging neurotransmitter release with Inscopix miniscopes. Just by means of a very brief introduction, obviously neurotransmission is foundational to the function of the nervous system, and it's a common therapeutic target for human and neurological neurological and psychiatric diseases, rather. And common examples would be using L-DOPA to treat Parkinson's disease or SSRIs for depression, stuff like that. And precise measurement of neurotransmission in behaviorally relevant contexts will certainly advance our understanding of brain function, as Albin's results kind of hinted at, and hopefully will lead to more effective therapeutics down the line. So genetically encoded fluorescent neurotransmitter sensors have been developed in recent years and have emerged as powerful new tools for monitoring neural activity. And published work with them so far have been mostly imaged in vivo using either 2-photon microscopes, which require head fixation and don't allow for free behavior of the test subject, or with fibrophotometry, which does allow for free behavior but lacks spatial resolution. So we've been working to utilize the advantages of miniscopes along with these sensors, which include being lightweight and head mountable to allow for free behavior and even allow for multicolor imaging of multiple indicators, including GCAMP with single cell resolution. I do want to briefly give the Inscopix miniscope platform a proper introduction, just so that everyone's familiar. Inscopix has 3 miniscope designs for various experimental applications. The Invista system allows for single channel imaging of green dynamic indicators, such as the calcium dynamic calcium activity movie that you see on the right side of the screen. Invoke also allows for imaging of green indicators and is additionally equipped with an LED designed for stimulation of red shifted opsins. So it allows for optogenetic manipulation of neural networks in the field of view. And we've recently released the InView system, which allows for simultaneous imaging of multiple indicators, including green and red dynamic indicators simultaneously. These systems are paired with implantable GRIN lenses, which allow for targeting of cortical, subcortical, and deep brain regions. So as we began our work exploring the use of neurotransmitter sensors with Inscopix miniscopes, we devised a simple experiment very similar to what Albin has already presented, so I will move through quickly. But we also used the green version of GrabDA that Albin also was using, and use it in this case in the dorsal striatum, and implanted a GRIN lens in the target region and used an Invista system to image the sensor's activity. Like Albin, we recorded for a few minutes of baseline activity and then administered amphetamine to induce a change in dopamine transmission to see whether we could measure it, and recorded for 15 minutes after the injection as the drug took effect in the test subject. And we're looking here at the results. These are mean traces of delta over F activity in the cohort of mice that received either amphetamine or a saline injection. You can see that in the saline trace, there's a gentle decrease in fluorescence for the subjects that received the just the control vehicle injection. And this decrease in fluorescence is most likely due to photo bleaching of the sensor itself. Whereas in the amphetamine trace, there's a consistent increase in fluorescence after the injection. And just so you can visualize how the actual miniscope data looks, this is an example image from the baseline period of a mouse that received an amphetamine injection before the injection was given, with a region of interest drawn around the area of the signal. And then here's an image from the same mouse 15 minutes after amphetamine was administered, and you can see a clear increase in fluorescence. We also verified that we could use Invoke to evoke dopamine release and visualize the response of the sensor. So again, similarly injected viral construct of GrabDA in the same brain region, and also used the excitatory redshifted opsin crimson R in the substantia nigra to infect dopaminergic terminals to allow for optogenetic stimulation of dopaminergic terminals in the field of view to directly evoke dopamine release. And we found that we were able to evoke a consistent time-locked response of the sensor to optogenetic stimulation in this case. But what I especially want to highlight is the capability of the InView system, which again allows for simultaneous imaging of green and red dynamic indicators. So we wanted to examine the possibility of using a neurotransmitter sensor alongside a dynamic calcium indicator like GCamp. So in this case, we used a red variant of the GrabDA sensor together with GCamp in the dorsal striatum, and then used the InView system to simultaneously image both signals. And similar to our Invista experiment, we administered amphetamine to induce a change in dopamine transmission. And as our example video here shows, we found that we were able to simultaneously measure distinct signals from each indicator very well. The way the InView enables simultaneous imaging of two indicators is by precisely alternating its two excitation LEDs as it gathers frames, such that every other frame contains a signal from one indicator or the other. As the recordings are then processed, the frames are sorted into two separate movies, which contains only frames either from the green or the red channel. And from there, each movie can be processed and analyzed, down the pipeline. In this case, the data from the green channel was processed like a normal GCamp data set. Cells were positively identified using a CNMFE algorithm, and then individual activity traces were extracted. And in the red channel, we use a large ROI to measure changes in the activity of the dopamine sensor. Looking at the calcium event data from GCamp in the green channel, in this case, we found we were able to identify as many as 250 individual cells when recording the data in this way. Each cell had its delta F over F activity processed with an event detection algorithm to look at calcium transients in an individual manner for each cell. And we can look at the overall changes as the time course of the experiment continues. You'll notice that in this experiment, the recording is not continuous, but it's divided into 11 discrete blocks. And so the reason we did this, you may recall in the Invista experiment that I presented, we saw a slight decrease in fluorescence in control mice, which was due to photobleaching. And so in these experiments, we were using a red dynamic indicator with the dopamine sensor. And with red indicators of all kinds, not just for this particular dopamine sensor, photobleaching is much more prominent. The red indicators are much more susceptible to photobleaching. So we use a recording schedule of 30 seconds on, 90 seconds off. So that we were still recording for about 20 minutes in total but the overall recording time was reduced. So this just allowed us to reduce the overall exposure of the red indicator to the LED to make sure that photo bleaching didn't overpower our signal. So looking at that signal from the dopamine sensor, we plotted those 11 recording blocks continuously here just to visualize the changes more easily. And similar to what we saw with the green version of GrabDA that I presented, we see a consistent increase in fluorescence again after amphetamine administration. The overall delta F over F is about half of what we saw, about half the amplitude compared to the DA2M variant of GrabDA. But this is expected based on the work from Yulong Li's lab, who developed these Grab sensors. And they characterized these two sensors and showed that in response to the same known concentration of dopamine, the RDA1M variant had about half as much change in fluorescence as the DA2M variant. So this lower amplitude is a property of the sensors themselves. So, in summary, we see that the eView Miniscope is well suited to be able to extract meaningful signals from both channels when they're recorded simultaneously. And that Miniscopes offer several advantages over other imaging modalities for these types of sensors, including being able to do multicolor imaging with high spatial resolution. They can be used along with the optogenetic manipulation of neurotransmitter release. And as always, they're lightweight and head mountable to allow for free behavior of test subjects, which is very important for many brain circuits. And simultaneous imaging, in particular of neural network activity alongside neurotransmitter sensors during free behavior, is totally unprecedented and will allow researchers like Alban to take their work even further than before. And this possibilities are especially exciting considering the availability of many other types of neurotransmitter sensors besides dopamine sensors. So we're excited to see what the field will do with these new tools. So yeah, just in summary, with Inscopix Miniscopes, you're able to record cell type specific neuronal activity over many months. Can visualize fast genetically encoded optical sensors of neurotransmitter activity and simultaneously investigate neural activity and neurotransmitter dynamics during free behavior. So thank you very much. Please let me know if you have any questions for either me or Alban.
Thank you, Kevin. I really appreciate both you and Alban, your efforts today. This was a fantastic presentation. I encourage everyone who's attending this webinar to submit their questions via the Q&A box. I would like to begin with the following question. We have a few for you. Alban, have you looked at the correlation between GrabDA dynamics and mouse behavior? For example, during locomotion or a reward task?
No, not yet. In fact, it's, as I said, ongoing work on that. But for the moment, the first result is try to see. And we are now, for the moment, we analyze some model of psychiatric disease to see exactly indeed if we find correlation between specific behavior with the release of dopamine or the dopamine transient. This is we are analyzing the result for the moment. The second don't give a definitive answer about that.
Mm-hmm. Excellent. And another question for Alban. Have you tried a D1 scaffold DA sensor, such as D-Lite 1.2 in the dorsal striatum? Would you expect similar spatial temporal patterns in baseline condition as well as post-amphetamine injection?
In fact, we when we both tried, our first imaging was better with GrabDA. This is why we choose it. But we have some, when we reanalyze some data from the D-Light, it seems, but we don't go deeper in the analysis of D-Light result. But for the beginning, the result was clearly more convincing or strong with GrabDA sensor than D-Light. But we don't go deeper in the analysis of D-Light because in our hands, GrabDA seems more exciting or more convincing.
Okay. I have another question here, Alban. Was the I guess there needs to be some clarity. Was the recording in the dorsolateral striatum or the dorsal medial striatum?
You mean from which one? The calcium imaging?
Yes.
Or no, calcium, but you know the size of the lens. Thus, we are to specifically target DLS or DMS is not so, it's not so easy.
Yeah, exactly.
Thus I can not clearly say, okay, this is DLS neurons or this is DMS neurons due to the size of the green lens.
Correct. Okay. And then let me see here. I'm getting a flurry of questions for you. How about how can you make sure if the fluorescence change is due to neurotransmitter release, but not the fluorescent artifact when you activate crimson? I think this might be a question for you, Kevin.
Sure. Yeah. So you did negative controls in these mice, in mice that had the dopamine sensor but no crimson, and still, recorded the dopamine activity and exposed the field of view to the optogenetic LED and did not see an increase. Actually, I think I have that on a slide that I can show you.
Absolutely.
Briefly.
Please feel free.
So if you look on this slide in the bottom middle panel, we're looking here at the fluorescent activity of these negative control mice. These had the DA2M sensor expressed, but no crimson. We activate the LED and don't see a response of the sensor to the optogenetic LED. So that's how we controlled for that.
Okay.
I can add something. The fact that when you use Haldon, the fact that you lose the transient is also an additional indication about the fact that it's not an artifact.
Right. Another question for Kevin is, I guess this might have been answered. Oh, let me see. Given the photo bleaching issue in the red channel, is there an explicit recovery curve defined for that yet? Your example gave a 30-90 on off, but could you do 60-180 or 10-30? Benefits or drawbacks to a longer on time?
So photo bleaching is persistent, so you would expect to see, I mean, not totally persistent, but on the time course that we're talking about, it's not like photo switching, which is kind of a different phenomenon that's quickly sometimes reversible. But photo bleaching will last for a period of at least a few days. So I think over the time course of a typical imaging session where you're talking, 30 minutes, an hour or whatever, what matters more is just the total exposure time to the LED. So having a longer on time, it will just be directly correlated to the degree of photo bleaching that there is. So if you record for 60 seconds at a time over the course of 20 minutes for 5 minutes of total recording time, it doesn't really matter so much whether you're doing 30 seconds 10 times or 60 seconds 5 times. The total photo bleaching will still be similar over the course of the experiment. It's just a matter of what kind of sampling frequency you want. So I've tried several of these things. And in our case, we found that we wanted to do a little bit shorter recording, a little bit more frequently, just so that we could visualize the time course of the change a little bit more easily. but yeah, I hope that answered your question.
Question here, I think for, I believe this could probably go to both of you, but do you manage to get a GrabDA fluorescence across the entire field of view, or do you need to select a specific region that is in focus?
Yeah, I can answer that one. I think it does generally, in my experience, fluoresce across the entire field of view. And because we did a rather large viral injection of around 700 nanoliters of virus in our case, the expression is rather broad. So you can see fluorescence from the entire field of view in general. And it seems to all essentially be in focus. I don't know, Albin, do you have anything to add to that?
In our case, we analyzed the image and so on generally in the central field of the full size field.
Okay. I have another question here, and this is probably for both of you. Fiber photometry signals can be obscured by motion artifacts, and many labs use a static TDT tag to provide a fluorescent control. It doesn't seem to be possible with the InView application. Do you have any concerns about motion artifacts using the miniscopes?
Yeah, so we don't offer motion correction in the same way that fiber photometry typically approaches it. But we are able to apply motion correction in the same way that we would for calcium imaging data. So because we actually do have spatial resolution with the Grin lenses, we're able to visualize landmarks in the field of view and stuff like that can be used for a motion correction algorithm, so that we can at least correct for like X and Y kind of translations, horizontal translations of the field of view to the extent that they're there. So that's one way that we're able to correct for motion artifacts. I'm not particularly concerned about our motion artifacts in the Z axis. I think just because the axial resolution of the system is rather broad, I don't think it would cause very disruptive changes in the fluorescence signal like you might see with fiber photometry.
Overall, I have a number of questions, just a number of commentaries from people in the attendance saying this was a great talk. Everyone is very pleased with the presentations today. I really want to thank you all. Overall, thank you so much. I see a fantastic set of questions here that everyone has brought in. And again, I want to thank you all for your time today. Thank everyone who attended. I appreciate everyone's time today. Alban, Kevin, it was wonderful hosting you today on our webinar. I want to thank everyone who joined us today, and I hope you found this information interesting, exciting, and insightful. Please stay tuned for future webinars from Inscopix. Feel free to reach out to us on our contact page at www.inscopix.com. I'm going to close out this webinar today, and I want everyone to have a wonderful holiday, and we will see you all in the new year. Thank you all for attending today. This webinar will now end. Salud.
Bye.
Thanks, everyone.
Bye-bye. Thank you.